Thank you for joining me in my presentation. And I would talk about, expand on what Professor Akbar Shah already talked about species difference, why we should use human in vitro systems. Then I'll talk about hepatocytes, my favorite system. I'll introduce you some of the newest advancement in hepatocyte technologies, and then, of course, the IdMOC system. I think that the concept that animals can predict human toxicity is a overly simplified concept developed almost 100 years ago. We now know that it doesn't really work that well, as indicated by the low clinical success rate. When a drug company, when the pharmaceutical industry develop a new drug, right, then we go through all the preclinical testing, including multiple animal species, and then they would choose the most efficacious and safest drug molecule that they hope that will be equally efficacious and safe in human. But that doesn't work, as indicated by this statistics. The drug candidates that has been chosen using animal studies have a 90% failure rate. 9 out of 10 drug candidates, when they enter clinical trial, would fail. The overall success rate is around 11%. So that really is the major, is very important data, illustrating to us that we need to do something better. So this is the conclusion. In spite of extensive preclinical evaluation, using at least 2 animal species, doing all the research work, the average clinical success rate of the chosen drug candidate in human clinical trial is only 11%. Major reason for the failure is unexpected adverse drug effects, toxicity, and then of course, lack of efficacy. And not only that, the very last bullet at the bottom here, frequently lots of drugs are withdrawn after they enter the market when human patients have been exposed to the drug. When a large human population of human patients are exposed, the drug will show serious toxicity, leading to the withdrawal of the drug from the market. Very simple. Clinical trial fails. Drugs have to be withdrawn from the market because of the lack of efficacy and because of the presence of adverse drug effects not predicted by animals. So the whole concept of using animals to predict human drug properties is flawed. We now know we have to do something better. I think that we are all very concerned that too many animals are being used unnecessarily for research and in drug development. This is a major problem. One of the reasons that animals are different from human is in drug metabolism. An animal species who does not metabolize a parent drug, the form metabolites found in human may under or overestimate drug toxicity in human. The animal would underestimate toxicity if it doesn't make the human toxic metabolites, right? If they make metabolites that are toxic only to human, it would overestimate toxicity if the toxic metabolites are only toxic to the animals but not to human. in both cases, totally, it's a total waste of the use of the animals. They have been sacrificed to yield no useful information. Scientific basis for species difference is well established. The major enzymes responsible for drug metabolism are the enzymes within the P450 family. And the major isoforms are 1A, 2A, 2B, 2C, 2D, 2E, and 3A. We know that. And look at that. All the ones in the red are the enzymes that are present in animals that are different from that in human. Different proteins. So therefore, different activities. So therefore, will form metabolites much different than that in human. There are only two P450 isoforms that are conserved among the species. That's 1A, that's mainly responsible for the metabolism of the fire chemicals are produced by burning organic materials like the aerial hydrocarbons. And 2E, the enzyme responsible to metabolize tiny chemicals like alcohol. So if you smoke and drink, probably animals are good models for humans, but for all the other drugs, almost animal species would make metabolites like human. And this difference is not just quantitative, meaning that, okay, the animals will make a metabolite that are found in humans, just maybe in a smaller quantity. It can be black and white. As you can see in this publication that was published in our lab many years ago, coumarin 7 hydroxylation, this one pathway, turning the drug coumarin to 7-hydroxycoumarin. It does not occur in the rat at all. It only forms in human. So the rat makes none of this, 0. Yeah. So that's why, and if the wrong animal species will give you the wrong results in human. Give you a real life picture. This is something we published a long time ago. This is the first author is a different Dr. Lee. It's not me. It's a scientist in a Belgium laboratory that I collaborate with. And the chemical in the center is the drug. And you can see that the human metabolites, you can see the human arrow here, forms metabolites, which we call HMs, right? Any M is a metabolite, H represent human, and R represent the rat. You can see that there are many rat-specific metabolites, many RMs, which are unique from the human metabolite, HMs. So for this drug molecule, for instance, the rat is a really, really not a terrible model for human. But toxicity, I think that India has been exposed to this chemical TCDD. Some of you may remember there's a faux pas incident. Union Carbide, the chemical company, there's an explosion, and human has been covered from head to toe with this chemical dioxin, TCDD dioxin. And this is a very toxic, extremely toxic chemical. If you are a guinea pig, yeah, you can see, if you look in the graph here, guinea pig, right? Look for guinea pig. The LD50 is around 1 microgram per kilogram. 1 microgram per kilogram. Very toxic. And then you can look for all the different animal species here. And when you see the word, you see hamster, right? Hamster is just a tiny version of a guinea pig. It's a pet, it's a house pet, and many of you have that as a pet in your family for your children. The hamster, the LD50, the dose to kill half the animals is 1,000. You can see that there's a 1,000-fold in toxicity. So this really tells you that it'll be very difficult if you want to use one animal species to predict toxicity of a chemical in a different animal species. You can be totally wrong. Yeah, you can make judgments that are totally irrelevant. How do you solve this problem? Again, Dr. Abasher has explained this a little bit in the introduction. We really need to have an experimental system that has the human specific properties, human pharmacology, human drug metabolism, human toxicology. If you have such a system, then you'll be able to predict human drug properties, especially efficacy and safety, much, much better. So this is a concept that I advanced many, many years ago, and is now being universally adopted. Many drug companies now apply human in vitro systems to predict human drug properties before they embark on animal studies and then, of course, clinical trial. The concept is really simple. You develop human experimental systems to obtain human-specific information, but you have to use the right system. Okay? You have to use the right system. If you use the right system, then you can choose the right animal species that act like human, and then you can obtain results that are much more relevant to human in vivo. What I'm proposing here is the reduction of the use of animals. Eliminate using animals for your specific question. Eliminate the species that would not give you humans results. That is a total waste of the use of animals. It's not proper. It's inhumane. It's a waste of energy and resources. If you can choose the right animal species, then you can use in vitro system to predict human in vivo properties of what you're interested in. You can either go directly from in vitro to in vivo using computer software that can allow you to do the in vitro in vivo correlation. That is available for some of the drug properties like metabolism, like pharmacokinetics. And you can use in vitro systems, human in vitro systems versus animal in vitro system to choose the most appropriate animal species for in vivo study that then can give you information that is useful to humans. Again, what we're doing, if we use this approach, then we'll be successful in predicting human drug properties, and most importantly, for the society, for all the efforts by different different philosophers and scientists and laymen who are concerned with the inappropriate use of animals, eliminate the use of animals inappropriately to provide results that are not useful. So therefore, reduce our use of animals. How do you define a good in vitro system? Real simple. Whatever you're using, it has to have the properties reflecting the organ that you're interested in. And then, of course, preserving the species difference, the species specific properties you are interested in, right? Use human. If we use human cells, make sure that the human cell have the organ specific property. And what are the key properties? This is what I call the MTE requirement. Your in vitro system has to have the right drug metabolism capacity specific to your species of interest. Like, for instance, human. It has to contain the relevant toxicological target if you're interested in toxicity. And then you just have to do the right experiment using the relevant endpoints. Let's talk about M. There are many phases of many pathways that are critical to metabolism. There's something called phase 0, which is uptake of the chemicals into the liver or the other organs of interest. Phase 1, that's the oxidation of the molecule, very active in the liver. Phase 2, conjugation of the molecule. Lots of chemicals are firstly oxidized and then conjugated and then excreted. Excretion will occur just by going through the membrane by permeability, and also they can be excreted via efflux transport. So phase 0, phase 1, phase 2, phase 3 are all considered metabolism. And then, of course, T, toxicity. So hepatocyte is a very useful model to understand hepatotoxicity, right? Because it has the right metabolism and it is the right cell type that's being damaged in the liver in vivo, leading to liver failure. So in my, in the many decades of my career, I've been lucky enough to stay and work with hepatocytes non-stop. And one thing I discovered very early on is that hepatocytes, when I started using hepatocytes, everyone would be isolating hepatocytes from an animal and then use them as soon as they're isolated for experiment. Number one, right, is very inefficient. You have to sacrifice an animal to make the hepatocyte, and you use a small portion of it, and then you throw away the rest. You cannot store them. And every single time you go and do an experiment, you would kill animals unnecessarily. I wanted to eliminate this, so I invented cryopreservation. So I was one of the first scientists to successfully cryopreserve hepatocytes. If you can make hepatocytes, cryopreserve them, then you can use them. First of all, there's no more wastage, right? Everything you make can be, whatever you don't use, you store in cryopreservation, and then you can do your experiment whenever you want just by thawing the hepatocytes. And this is especially important for human hepatocytes because human livers are really difficult to come by. Right? With animals, you can purchase an animal and use them at your ease. But if you want to do experiments with human hepatocyte, you have to wait until a liver is available. Livers are procured by surgeons for transplantation, and they are available for research if they cannot find a match donor. If the liver cannot be used, then they are available to be used for research. So they're very rare, very difficult to get. Cryopreservation is the only way to make the experimental system useful and available universally. So you can see that this is the different stages of our progress in the use of human hepatocytes and animal hepatocytes. In the late 80s, we firstly demonstrated successful cryopreservation. And then in the late 90s, 10 years later, I was with a company and we were able to successfully demonstrate cryopreservation to retain metabolism and as well as providing cryopreserved human hepatocyte as a reagent worldwide. And then I worked with lots of international experts to talk through the system, to do experiments with the system to show the usefulness. In the early 2000s, we demonstrated that hepatocytes are not only metabolically active, they are also active in the uptake in transport, in the transporter activity. And then the last bullet is very important. We show that we can culture the human hepatocytes. Before 2007, hepatocytes after cryopreservation were considered not culturable. They can only be used in suspension and they survive only for hours. So I discovered this, I invented this recovery medium. It's now used universally. It's also called CHRM, CHRM. It's a medium that we manufacture for GIPCO to sell to hepatocyte researchers. You can see that the viability using CHRM is on average is around 90%. 90% of the hepatocytes are viable after cryopreservation. And this is what we show when we compare activity of the fresh hepatocytes versus the cryopreserved hepatocyte. You can see that all the lines with the slope of unity is a 1-to-1 correlation. And many scientists now, not from a laboratory, use hepatocytes for research. And this is just an example on the publications. And the very last paper here, the highlighted paper, is the first demonstration that hepatocytes, cryopreserved hepatocytes, are functional in efflux transport. They can actively transport chemicals actively away from the liver. So hepatocytes are totally capable of phase 0, which is uptake, and then phase 1 and phase 2, which is oxidation and conjugation, metabolism, and then of course phase 3 is efflux transport. All right, let's talk about plateability of hepatocytes. Why do we want hepatocytes to be culturable? Number one, if they cannot be cultured, the hepatocytes, fresh hepatocytes, can be cultured really well. Cryopreserved hepatocytes, because of membrane damage, that's why at the beginning, they cannot be cultured. And then, if they cannot be cultured, they're only alive for hours. So they can only be used for uptake study, metabolism studies, but they cannot be used for many other applications that are important, like, for instance, long-term metabolism, toxicity, especially metabolism-dependent toxicity. All right, I'm going to tell you our latest progress in culturing cryopreserved hepatocytes. Is what we call 999 elite hepatocytes. It's something that we are very proud of. Yeah, we now have near perfection of human hepatocytes. You can see that this is one of the lots of hepatocytes. You can see that you plate them on day 0, they form beautiful cells. Almost all of them with plate. Yeah, almost 100% plateability. You can see that the cells can be cultured for 40 days in culture. The morphology shift a little bit with longer and longer time in culture, but they stay viable for 40 days. Not only that we can do that, we have improved our cryopreservation procedure now that we can even culture cryopreserve hepatocytes twice to make the pool donor hepatocytes. This is our procedure. Yeah, pool donor meaning that you take hepatocytes that have been cryopreserved from individual donor, right? Each time you get a liver, you make hepatocytes. We can now cryopreserve them so that they can be platable, right, for long term, which is what I talked about. We can also pool them, which means that we'll thaw the hepatocytes after cryopreservation from different donors, mix them together. So now with a mixture of hepatocytes from multiple donors. So one suspension will have hepatocytes from different donors. And then we'll re-cryopreserve them, and we have a patent on it. And they also are platable. And that is very important. Now you can use hepatocytes, use them pool donor hepatocytes for research to give you information representing multiple individuals, so therefore can be what we call general properties of a general human drug properties, eliminating or at least reducing individual variation. Now, both are important. You need to know how different individuals are different in drug properties, because the drug you make will be exposed to different human populations. Individual differences are very important. But then for general understanding of drug properties, you can use pool hepatocytes to give you an idea of what is the overall average property of a drug. Okay, so now let's talk about application of hepatocytes, right? Remember what I talked about, the phase 0 metabolism, uptake, transport. I want to show you that definitely hepatocytes are capable of uptake transport. So pravastatin, as shown here, is a substrate of uptake transporter. When a drug is uptaken by transporter, so there's a phenomenon called substrate saturation of transport, right? When the uptake is depending on activity of an enzyme, in this case is a transporter, then you can saturate the rate. Eventually, all the transporters will be occupied. So you can only the rate of transport will plateau, and that is demonstrated here with the blue dots. You can see that, right? When we look at rate versus concentration, there's the line curves down, demonstrating saturation, and that is very important to demonstrate that the activity is enzyme driven. Saturation kinetics is the basis for all enzyme kinetics, and same for transport of drugs. And when you put in a uptake transporter inhibitor, the orange dots here, that's an inhibitor called rifampin. You can see that uptake is reduced. So using hepatocyte would definitely have uptake transport demonstrated by the saturation of the uptake by high concentration of the drug, as well as demonstration of the inhibition of the uptake by a transporter inhibitor. Yeah, so every single one of our platable 999 elite hepatocytes are active in uptake transport, demonstrated by the inhibition of transport by the inhibitor. You can see that all of them are inhibitable by rifampin, the inhibitor. Enzyme induction. Enzyme induction is a very important criteria of drug interaction. It's required for new drug application, NDA, new drug application. Every drug has to obtain to be successful, to be accepted by FDA and provided with this NDA, new drug application. And before FDA allows you to market the drug, to give you the NDA, you need to show that the drug does not have significant drug interaction. And FDA requires for you to understand if your drug can induce drug metabolism. Imagine what happens, right? If you take a drug that induces drug metabolism, you after taking the drug, your body will eliminate drug much faster. If you take another drug with this drug that induces metabolism, which is metabolized by the enzymes that are induced, then the other drug will be eliminated much faster and you would lose efficacy. And this happens a lot with, like for instance, cyclosporin, which is important for people who have been transplanted an organ. It's an immunosuppressant to prevent organ rejection. But if you take an enzyme inducer, like rifampin, then the patient would reject the organ. The cyclosporin would no longer be effective because it's metabolized much faster. And so this is a very important criteria. So US FDA and many other regulatory agencies says you have to look at induction to make sure that it doesn't induce the key isoforms that are inducible, 1A, 2B, 6, and 3, 4. And if it's inducible, then you have to look at the other isoforms too, the 2Cs, 2C8, 2C9, 2C19. So therefore, you have to have hepatocytes that are inducible. Yeah. So our beautiful hepatocytes, are they inducible? That's the question. And of course they are. You can see that there are two ways to measure induction, measure gene expression, measure activity of the enzyme. You can see dose-dependent increase in activity in messenger RNA for 1A2, 2B6, 2E4, those major isoforms that are inducible. And then, of course, 2C8, 2C9, 2C19, the other isoforms that are also inducible. So our hepatocytes, our beautiful, highly culturable hepatocytes are all inducible for all these isoforms. Right, I talk about pool hepatocyte pool of multiple donors. They are also inducible for all those isoforms. Toxicity, right? We're all interested in toxicity. We have developed many assays for toxicity. I'll go through some of the easy ones, right? This is the easy one. Just treat with drugs using your culturable hepatocytes, and then a day or 2 days, 3 days later, measure viability. Usually we use ATP as our endpoint, but you can also use enzyme release like LDH or AST, ALT. You can also measure MTT, mitochondrial metabolism. On top, you can see that estradiol, the non-toxic estrogen, is not hepatotoxic. Tamoxifen, the hepatotoxic estrogen analog, big difference in toxicity, and hepatocytes can tell them apart. At the bottom, there are two drugs known to be different in liver toxicity. Troglyderazone, the red, is a lot more toxic than rosiglitazone, the blue. And this is also true in vivo. What data shows us is that we can predict human liver toxicity using hepatocyte in vitro. And you can use hepatocyte to ask the question, which animal species is more like human in toxicity? Or which animals are not like human in toxicity, so therefore it's not a good model for human. On the left-hand side, you have cyclophosphamide. You can see that, right? There are many lines here, meaning that every line is one animal species, lots of species difference. The interesting thing is the black line, that's human. Human is a lot more resistant to the liver toxicity effect of cyclophosphamide. Rodents and dogs are a lot more sensitive to the toxicity. On the other hand, on the right-hand side, Tacrin. Yep, Tacrin is a drug that has been marketed and then withdrawn because it caused liver failure in humans. The black line, that's human. You can see that it's much sharper. Right? It goes down much faster than all other animal species except the monkey. The rat, the mouse, the dog are resistant to tacrin toxicity. This is a very clear illustration why in the old days, when you look at toxicity using animals, most popular are mouse, rat, and dog. They do not predict the toxicity of tacrin in humans, which is why when tacrin went to the human population, it caused liver failure and needed to be withdrawn from the market. But by using hepatocytes from multiple animal species, you now can choose the most appropriate animal species for in vivo toxicity studies. Because you can culture hepatocytes for a long time, you can also perform experiments by treating hepatocytes for a long period of time. In this case, in this experiment, we compare toxicity of drug after 2, 7, and 14-day treatment. You can see that in some cases, they're not much different to toxic upfront. And in some cases, the longer you treat, the more toxic is the chemical, and the survival curve shift left to the left-hand side. And this is an assay that we have developed. We hope to be able to predict drugs that induces liver failure, something what we call SDILI, severe drug-induced liver injuries. There are many drugs that went out to the market and then have been withdrawn because they cause liver failure, or they're very much limited in their use because of the severe toxicity, many of them. So because they have been marketed, so it's clear that you cannot identify these drug candidates before they are marketed using animals, right? The clinical studies, or even clinical trial. You cannot. So FDA, we collaborate with US FDA. They ask us if we can somehow come up with ways to identify these drugs. So we have a 3-year collaboration. We were lucky to be funded by US FDA for 3 years at around $100,000 per year. And they provide us with drugs. They're coded. We have no idea what they are, but they know whether they cause liver failure or not. Basically, they just give us toxic drugs, and then we provide it with the data. We have decided to use well, after we've done the research, I'll give you the summary. We discovered that ROS, reactive oxygen species, ATP, cellular ATP content, are the most useful endpoint, especially when you use the ratio, ROS ATP ratio, as the combined endpoint, and also especially when we express the data as the area under the curve. So when you plot ROS ATP ratio versus concentration, and then use the area under the curve of the dose response curve as the endpoint, we have the best information. Why? ROS reflects oxidative damage. ATP, the lower it is, the more toxic a drug is. Then the ratio of ROS over ATP is the combined effect of oxidative damage, and toxicity. it gives you a measurement that represents these two very important toxic endpoints. And then by using the area under the curve, you're defining the property of the drug at all the concentrations, rather than depending on one specific concentration, which is something that people do but unsuccessfully. How can you go from in vitro IC50 to in vivo toxicity, right? So we decided to look at the entire curve and use that as a property of the drug that you're studying. Again, to make a long story short, we discovered that using the RS-ATP ratio, using the AUC, area under the curve, as an endpoint with 90% sensitivity and 86% specificity. So that means that 9 out of 10 drugs that are toxic, that cause liver failure can be identified. 8.6 out of 10 drugs that do not cause liver failure, because the specificity can also be identified. So this is the best endpoint ever to identify these drugs that cause liver failure. And one thing is very, very good news to us who are interested in vitro systems. What if an in vitro assay that provide data that cannot be obtained using animal models or even in human in vivo? But our little cells in vitro give us useful information, very important information. Next application is in gene therapy. There are many diseases in the liver that can be overcome, but they can be treated not by chemicals, not by ordinary drugs, but by gene therapy. And this is a very rapidly developing field. Gene therapy is now finally real. These are many of the diseases are being investigated to be overcome with gene therapy. To treat liver diseases using gene therapy, you have to have a way to deliver the gene therapy modality into the liver. And the ways to do it is there are two major ways to do it. One is AAV, adeno-associated virus. AAV, just by experiment, is something that has been observed experimentally, goes mainly into the hepatocytes. For some reason, when you administer AAV in human, the liver picks them up. So therefore, it's a good vector to deliver a gene into hepatocytes. And the other is called GalNAc, GalNAc. Yeah, it's a glycoprotein, galactosamine-containing glycoprotein on the membrane of the hepatocytes. So if you deliver a drug by its binding to the GalNAc receptor, it will go mainly into the hepatocytes. So there's a way to deliver drug gene therapy modalities selectively into the hepatocytes. So when you try to develop gene therapy for human diseases that are in the liver right now, you have to ask the question, right? You're trying to overcome a human disease. So animal models are practically very difficult to be useful. So, what can you do? What can you do? So the one model that drug companies use is what they call humanized mouse. A mouse that, due to a specific genetic disorder, is the hepatocytes will die. Yeah, after a certain day, certain age. But if you transplant human hepatocytes into the mouse that survive would have the human hepatocyte taking over the liver. Those are called humanized mice. A very useful model, right? Now you have an animal with human hepatocytes, so therefore can be used for gene therapy research. Except it's very, very expensive and also it's inefficient, right? To study one drug, one concentration, one duration, you have to use one animal. Yeah, you have to sacrifice one animal that only represents one. If you want to use multiple animals to make sure that it's reproducible, you have to use multiple animals at each concentration, at each duration. So very expensive, and we are now working with drug therapy company, and this has now become almost routine. What we told them was use human hepatocytes. That's it. You can ask the question: does the drug get into the human hepatocytes? Does it work on the human genes like what you're expecting them to? Right? Real simple. And you can use 96-volt plates to a high throughput manner. So therefore, you can save lots of chemicals. You can do many experiments and many endpoints simultaneously, very, very efficiently. So we initiated a program, collaboration with Amgen in the United States, to ask the question, can we use hepatocyte to understand if gene therapy modality is efficacious? And ask the question, how long does it last? How long does the effect last? So this is recently published. We work with two Amgen scientists, and they provided us with a GalNAc siRNA. siRNA is the RNA that silences the expression, so inhibit the expression of a specific gene that they're made for. They use a model target gene, HPRT. HPRT, hypersensitive phosphorylysyl transferase, is present universally in all cells. And this, they want to ask the question, does the GalNAc siRNA inhibit this gene in human hepatocytes, inhibit the expression of this gene? So does it get inside hepatocytes? Does it inhibit the gene expression? And then how long does it last? We did the experiment. It's just the results are beautiful. After 24-hour treatment, right? We treat cells on day 0, and then 1 day later, remove all the medium, stop the treatment. We measure gene expression. You can see the arrow here represent HPRT gene expression. All the other genes are the albumin, ALB, TF transferrin, TTR transthyretin, and ASGR1. That's the gene for the receptor that allows the binding of the GalNAc siRNA to allow them to be uptaken into hepatocytes. You can see that the HPRT1 gene, the target gene, expression is totally suppressed, right? 80% suppressed after 1 day. And then the beautiful observation is that the suppression effect lasts for 33 days. One treatment, gene expression is decreased over 80% for 33 days, and then then it loses its effect after 41 days. So this is the first application of hepatocytes demonstrating that siRNA effect, the gene suppression effect, after one treatment can last for a month. So that's very useful information. Another application of hepatocytes is in the treatment of fatty liver disease. It's a real important area. Many people suffer this disease. And many people suffer it because they carry the wrong gene. There's the PNPLA3 gene. You don't have to know what it is, except that it's a gene that's responsible for chronic liver diseases. With this gene, if you have the GG genotype, then you have severe obesity and you have loss of fat in your stomach. Yeah, and this disease, this fatty liver disease, can progress to liver failure. So we look at our hepatocytes. You can see that with many of these we genotype our hepatocyte, and we identify the GG genotype in one of the lots, right? HH1072, the big GG letter right there. We have that. So that's very exciting. So now these hepatocytes are being used in fatty liver research by several drug companies. And this, the MBOAT7. This is a liver hepatocyte genotype that's important. The TT genotype is found to be associated with increased risk of fibrosis after HCV infection. So we ask the question, do we have hepatocyte with TT genotype? And we do, yeah. The same, yeah, 1072 is also have the TT, MBOAT TT genotype, as well as 1122, as well as 1140. And this is the another fatty liver disease gene that's important. GCKR gene. Two genotypes that's important. The CT, the heterozygous genotype, yeah, they're associated with low HDL-C levels, right? HDL, high density lipoprotein, is high density lipid. It's good. You want to have high HDL, and those people have low HDL. And if you have the TT, homozygous mutation, then you have excess risk of many metabolic diseases, including insulin resistance, elevated blood pressure, and so on. And we also have the TT as well as CT genotype in our hepatocyte bank. Yeah. I think that most of you, right, in vitro cell culture, you understand the 3D culture of cells is a big field of activity. Our hepatocyte also forms spheroids. This is one of the lots. You can see that it goes from a loose aggregate to become a totally tight spheroid after around 3 to 7 days. And many of our hepatocytes, 999 hepatocyte, also form spheroids. So these are, I really think that these hepatocytes are just wonderful. Can be useful for all types of applications, for metabolism, for toxicology, for pharmacology, and also for 3D biology. And that's what we're doing right now. We're trying to develop long-term culture hepatocyte with fully functional activities. And just want to show you some of our progress. We're not there yet, but we're almost there. We show that hepatocytes, they really differentiate, right? This is expression versus time for different key hepatocyte genes: albumin, transferrin, transthyretin, ASGL1, the protein receptor, for GalNAc, siRNA, and then HPRT1 is just a normal undifferentiated marker. You can see that hepatocytes do differentiate after 2 days, but everything comes back after 7 days. Same for the P450s. Same for the uptake transporters. Same for efflux transporter, it doesn't really drop, but it mainly maintains all through. Yeah. So So we now have our 99 elite hepatocyte that we like very much. We have them from single donor and multiple donor. They're highly viable, highly culturable, many applications, and they also will re-differentiate after prolonged incubation. We have hepatocytes from multiple donors, from human, and also from all animal species. Okay, so this parallelogram, right, that I talked to you about before, you can use hepatocytes in this parallelogram to understand human in vivo liver drug properties. With liver, it's okay. But what if you want to look at effect on other organs, right? With the liver, you have metabolism. But if you look at toxicity, like for instance, of the heart, you use cardiomyocytes, then you're only looking at the toxicity of the drug itself without the liver, without metabolism. So you won't have to write information about in vivo drug toxicity. So I want to come up with a system and to allow organs to be co-cultured with the liver to allow human drug toxicity to be evaluated not only in the organ of interest, the non-liver organ of interest, but also in the presence of liver metabolism. So this is the concept of the IdMOC model, IdMOC model. We are an organism with multiple organs that are physically separated or discrete, but they're connected by the blood, right? So integrated. When you integrate it to the liver, then you can have liver metabolism that will allow liver metabolism to be a factor in the property of the drug that you're studying. So we take an ordinary 6-well plate, as indicated here. We change it, so we lower the barrier. Now you can see that, right? By flooding all the wells, they're all connected by this overlying medium. So if the center well here is the hepatocyte, a drug can be metabolized by the hepatocyte. The metabolites can now go out and cause toxicity in cells in a different well. So therefore, telling you effect of the metabolite on that in that well that physically do not contain hepatocyte, but now is affected by the metabolites of the hepatocytes. So that's the IdMOC concept. Right? That's the actual plate. It's a 24-well plate with 4 compartments. So you can actually do 4, n equals to 4 for each treatment. And on top is how you load each well. I think that if I were there with you guys in a workshop, you will see this display and you'll learn how to add the cells to the individual wells. And then after the cells are cultured, then you just add extra medium, which is the bottom right well. You can see that you have medium covering all the wells. So all the cells are now in communication to the medium covering them, right? So it's a real useful concept. And we also have a 96-well plate model. I think most people use this model that's compatible using multi-channel pipettes. So this is the IdMOC. It's a wells in a well concept, allowing co-culturing of multiple cell types as physically separated entities, but interconnected by common overlying medium. These are some of the publications. I'm going to show you how well it works. Again, if you were working physically in the workshop, you would do this experiment. We use IdMOC model, and this is the 4 chambers, right? First one, we use just the 3T3 cells. And then we add 1, the yellow, the 2, and 3, co-culture with 1 or 2 or 3 wells with hepatocytes. 3T3 cells, right, cannot metabolize. Hepatocytes are metabolically competent, so now you're adding more and more hepatic metabolism to the IdMOC. And you can see that with no hepatocyte, the blue, you don't see any blue bars here because there's no metabolism. 1 well, 2 well, 3 wells, so different colors. The more hepatocytes, the more metabolism you have. So does it work? Can we show that cyclophosphamide is toxic? Cyclophosphamide require metabolism to be toxic, so we treat these 4 different co-culture systems, configurations with cyclophosphamide, and you can see that no hepatocytes, the blue bar, no toxicity. 1 well of hepatocytes, the brown, the dark brown color, very little toxicity. 2 wells, some toxicity, the gray bar, and then 3 wells, lots of toxicity. Yeah. And then these are 3 different plates, identical results. And in this experiment, we used MTT. MTT is a really good endpoint for IdMOC. It's because after treatment, you can just dump everything out, and the MTT crystals stay with each of the wells. Then you can solubilize the MTT crystals in each well, measure the color. And you can see here, right, no hepatocytes, the top, all the cells are blue, right? With 1 hepatocyte, and then you can see that all the cells are still blue. With 2 hepatocytes, then you can start to see some, the top line, the three wells. You can see that, like on the right hand side, you can see that the center well, all the 3T3s are dead, no color, and it's because that well is exposed to metabolism that is the closest to both of the wells of the hepatocytes. And then, of course, with 3 hepatocytes, everything is killed. Hepatocytes killed by cyclophosphamide because of metabolic formation of the toxic metabolite, but also the metabolites spill over and kill all the 3T3 cells. And by looking at the data, you can also classify how a drug how metabolism affects the toxicity of a drug. Tamoxifen is a drug that's directly acting. So it doesn't matter whether you have hepatocytes or not, same toxicity, pretty much so. Aflatoxin B1, it will kill the hepatocytes only. Not much toxicity to the 3T3 cells. It's because all the toxic metabolites are so active, it kills only the cells that make the metabolites. Right? Only kills the hepatocytes. So the gray bar is the lowest. 3T3, not much. Cyclophosphamide, just the opposite. Yeah, of course, it kills the hepatocyte that make it. It also can go out and kill the 3T3 cells that are co-cultured with the hepatocytes. Yeah. Without hepatocytes, cyclophosphamide is toxic only the highest concentration. With hepatocytes, the orange bar, you can see dose dependent decrease. Much higher toxicity than without hepatocytes. And the gray is a hepatocyte. Hepatocytes are also killed by cyclophosphamide. So you can now classify drug based on metabolism, drug toxicity based on metabolism. You have direct acting, no difference. You have type 2 that are metabolite. The drug is metabolically activated, but it only kills the cells that make the metabolites, right? The aflatoxin B1, type 2. And then type 3, a cyclophosphamide, it goes out and kill other cell types. The metabolite can come out of the liver and kill other cell types. This kind of classification is very useful, for instance, for anti-tumor drug development. You can also use IdMOC to understand organ-specific toxicity. Yeah, the spleen is a major organ for immune function, and cyclophosphamide is not only cytotoxic, it's extremely toxic to extremely toxic to spleen cell, to splenocyte. And this is done, an experiment done by Harry Harin when he was a postdoc in a laboratory. He's now a professor in Saudi Arabia. You can see that with only spleen, single culture, very little toxicity. If you co-culture hepatocyte, very toxic. Yeah, we're measuring. Harry measures the effect of PHA to stimulate splenocyte DNA synthesis and show that the response to PHA was totally abolished by co-treatment with hepatocyte and cyclophosphamide in a dose-dependent manner. You can always use IdMOC to understand organ-specific toxicity. Aflatoxin B1, when you so we have to study that we use hepatocyte in the center, RPTEC, which are the kidney cells, the renal proximal tubule epithelial cells, and SAAE-C, that's a small airway epithelial cells. Yeah. So we ask a question, hey, how toxic is something to each of the cell types? And with aflatoxin B1, a non-liver toxicant, you can see that it's mainly toxic to hepatocyte, the blue bar. Yeah, very little toxicity to the other cell type. There is toxicity, but not much. Yeah, bisphenol A, toxic to hepatocytes. Biphenate, yeah, there are lots of compounds that seem to hepatocyte-specific toxicity. Many compounds. Yeah. And you can always use IdMOC as a tumor-bearing man by putting tumor cell in there and then putting normal organ cells in the other world and co-culture them. This will be useful for chemotherapy and anti-tumor, anti-cancer drug development. To ask a question, can I come up with a drug that would only kill the cancer, but not the normal cell types? And we've done the experiment against tamoxifen, for instance, is the most toxic towards MCF7 cells on the right-hand side. Yeah, it is toxic, but less toxic to the other cell types. And then you can come up with the in vitro therapeutic index. You actually develop EC50, EC90, EC99 values. Like ask a question, which organ, what is the therapeutic index to each organ? Asking a question, how toxic is someone to is a drug to different cell types? In this case, you can see that this drug has a low therapeutic index to the endothelial cells and to the lung cells. Right. We have also studied nicotine toxicity, and we showed that it's interesting, isn't it? Yeah, look at that. Nicotine is really toxic to NHBE, normal human bronchial epithelial cell. Yeah, and what's interesting is that you can see that there is a increase in MTT in the other cell types. That means that there are more cells there after treatment with nicotine in the small airway epithelial cell, SAAE-C, and then microvesicular endothelial cell, the low concentration. And this is very interesting because that means that nicotine induces cell division for these specific cell types. Cell division is a pro-carcinogenic event. Yeah, when you're something that induces cell proliferation, you will promote cancer formation. So we have the IdMOC seems to allow understanding of tumor promotion also, which may explain why cigarette smoking is so carcinogenic. Not only that it has the carcinogenic components, right? All the polycyclic amino hydrocarbons, all the nitrosamine. It also can promote cancer formation. It also promotes cell division, therefore fixing the mutation that's been induced by the genotoxic components. All right. So the IdMOC is a practical in vitro system. Of course, we cannot co-culture all the organs, but we can co-culture some organs, the key organs, and it allows you to understand the role of hepatic metabolism in toxicity. Can be used to understand organ-specific toxicity. You can also understand cell type specific toxicity, as shown with the cigarette smoke condensate. It can be used to understand not only hepatic metabolism effect on drug toxicity to non-hepatic organ. It can also be used to as an experimental model for paracrine effects, right? What is the effect of the natural metabolites from one organ on the biological function of another organ? In the paracrine effect, you're actually looking at the effect of one cell type in an organ to another cell type in the same organ. For endocrine effect, looking at the effect of the metabolite from one organ on the biological function of a different organ. So I really believe that it's a very interesting experimental model, not just for toxicology, not just for metabolism, but also for endocrinology. So in this parallelogram, you use human in vitro to understand human in vivo. You use comparing animal in vitro, human in vitro to choose the right animal for in vivo studies to understand human in vivo drug properties. Right? With hepatocyte, understand the liver specific effect. Then with the IdMOC, you can understand, hopefully, the whole organ. And we've never done it because we're too busy, but we should do this. Maybe we can collaborate to do this. We now have made this IdMOC plate we can co-culture 96 wells. So we can actually put in all the organs and all the different cell types of each organ and co-culture them. So it's what I keep on calling it next generation. Hopefully it becomes this generation. Maybe we can set up a collaboration to study this. Is to have a human on a plate with all the key organs, so that we can study the effect of a substance on all the organs when they're all co-cultured together.